So I'm very pleased to be here to introduce Adrienne for her thesis proposal defense. As most of you probably know, Adrienne joined us from Dartmouth, and she wrote, she's probably sick of this story, an incredibly compelling essay in her application for graduate school about how much she loved working in mud and working in sediments and how great it was. And as you probably realize, this is a very rare type of perspective to have from someone. She had gotten her degree from Dartmouth, and then she had spent a year working for Ph.D. at Harvard, and she'd been to sea and had all sorts of adventures and was ready to go for more. And I thought, you know, someone who knows something about mud and really likes it is very rare, should be snapped up immediately. And it's been a great thing having Adrienne here. As you will notice for her PhD thesis proposal, her interests have evolved considerably. So she has indeed accomplished work with mud. Her first paper in that direction was published this spring. And she also did a very large project in the water column, which has also been submitted for publication. And now her interests have evolved in further directions dealing with this complex data that we get from experimental studies, where we're trying to connect all of these disparate parts in very difficult ways, and in many cases being frustrated and not being particularly effective at what we do. So Adrienne is taking a much bigger perspective on this problem that plagues us not only with mud and not only with the water column, but with data management in general. And she is coming up with some solutions that I think will have very broad-ranging implications. So take it away.
As Carol said, I'm really interested in how we can take really complex microbial data and try and derive the real functional significance from this data and use it to tie that to, say, biogeochemical patterns or environmental conditions on a global scale, and also to try and understand how microbial communities and the Earth have sort of co-evolved with the geochemical conditions on Earth. So in doing that, you end up having to deal with really large data sets, and you also run into pretty quickly a lot of computational challenges and data management challenges. So I'm also really interested in how we can come up with solutions to make this kind of large-scale comparative analysis easier for the broader scientific community, and of course for myself as well. So as we know, microbial communities have a really big effect on biogeochemical cycling. Virtually every biologically active element passes through a microbial filter. And this is just one sort of general schematic of how microbial communities impact CO2 fluxes on Earth. And I've highlighted the microbial bits in pink and then the reservoirs of carbon in blue. And you can see that microbial communities they're in terrestrial systems, they're in marine systems, they're in sedimentary systems, and they impact the amount and flow of carbon in all of these areas. For example, in soils, microbial necromass accounts for up to 80% of soil organic carbon, even though in those systems, most of the fixation of carbon isn't done by microbial systems themselves. So you have, that just shows how hugely microbial communities are transforming these systems. So the way this works is you have a pretty small community scale process, and they're taking in something and they're letting something out, and all these things interact so that when you go up to an ecosystem scale, that impacts the net flux of these elements through those through the community and through the ecosystem, and ultimately this impacts the reservoir of that element throughout Earth. The numbers are hard to see here, but they're the same as what we had on the previous slide, and that can really impact things like the net CO2 in the atmosphere, the amount of oxygen that we have to breathe. So we know from a lot of experimental evidence, we've documented really well this community scale process, and even starting to get up to the community and ecosystem scale fluxes, but we're really bad at trying to predict how all of these things that we can document in experiments and in the field actually interact with biogeochemical cycles, and how we can predict microbial components in the context of biogeochemical and functional capacities. So people have come at this question in a lot of different ways, and the takeaway from the slide is that none of them have been like particularly successful, but they've yielded some really interesting results. So we can see that microbial communities vary on a spatial scale, and this very reasonably could have global scale impacts on fluxes. People have tried to model net or sort of general richness and diversity metrics. They've tried to measure or tried to predict abundances of particular species. They've tried to predict particular genes, particular pathways. But none of this really seems to capture the actual functional significance of complex microbial communities. So the real crux of this, because of this, we haven't been very good at incorporating microbial processes that we know are really important into things like Earth system models. So what we currently do, if an Earth system model takes microbial information into account at all, which a lot of them don't, they'll assume some like really general stuff that we know is not true. They'll assume the carbon processing capabilities of all microbial communities are the same no matter where you are on Earth, and that the amount of activity that they're exhibiting has follows some sort of really general Michaelis-Menten kinetic rule with temperature. And we know that this isn't how it actually works in the environment. So we'd like to get closer to incorporating microbial information explicitly into system models so we can account for the spatial variability in these communities. So the real crux of the question is, how can we define the functional structure of microbial communities in a way that's actually relevant to the biogeochemical and ecological functions that they're impacting? So I would argue from this perspective that a microbial community is not a bag of genes that all have different annotations and these add up somehow to their biogeochemical function, but instead it's more useful to think of them as a mixture of latent ecological functions that are all present in different proportions, and that these together add up to the fluxes that we see. And then the functional biogeography that we see is the result of differential distribution of these functions across space. So the question is, how can we represent microbial communities that are really complex and really diverse in a functionally and biogeochemically significant way, especially when this information has a ton of genes. These communities are really diverse and complex, and there's a lot of functional redundancy in them. And where most of these genes, the interactions between them aren't really known, and this is particularly true because the vast majority of proteins aren't annotated. So we're really in need of new approaches to uncover the functional significance of complex microbial data. So the major research questions that I'm interested in for my dissertation are what the underlying functional structure, biogeochemical functional structure of microbial communities is, and how can we represent this? So how can we cluster genes, for example, into functionally relevant groups? And how can we use this to classify microbial communities into useful functional ecotypes, for example? And finally, given this functional structure that we've uncovered, what are the global patterns in microbial functional biogeography? So can microbial communities be predicted from environmental data? And if so, can we predict microbial communities through time with, say, environmental change? So in order to answer these really global scale questions, that obviously requires a global scale data set. And luckily for us, over the past 10 years, there's been a huge explosion in the amount of sequencing data that's available. And I am estimating, based on when I looked at this data, that there's now about 3,000 to 3,500 soil shotgun metagenomes that are in existence, and about 800 to 900 marine metagenomes. And I'll say that for this project at this point, I've chosen, because there's so many more soil samples than there are marine samples, and in my opinion, the comparative studies that have looked at soil metagenomes are more lacking than in marine systems. I've actually chosen to focus on soil metagenomes, but I'll be doing also a proof of concept in marine systems, and I can imagine applying it to marine systems, assuming this sort of works out. So anyway, all of this data represents a really vast resource and opportunity to ask these really global scale questions that can answer, fundamental questions of microbial organization. But it also, of course, presents a lot of computational challenges, both in how do we go about getting the sequence data. It's actually pretty hard to find sequencing data that meets whatever your specifications are, and integrating these into comprehensive comparative datasets. And then the computational challenges that arise when you're dealing with these really large-scale datasets that are petabytes of raw data. For example, when you do a small study where you have a couple of metagenomes that you're looking at, you can often assemble those metagenomes and compare your metagenome to your assembly pretty easily. But when you get up to the scale of 3,000 to 3,500 metagenomes, that becomes a much more difficult challenge. So you need some solutions to make that process easier as well. So to answer these two questions that I'm most interested in, I have a 4-part research plan. The first bit is that I need to get my global curated soil metagenome data set. And to do that, I'm also coupling this with development of a sequencing data discovery tool that I'm calling MetaSeq that'll facilitate integrative comparative analyses throughout the bioinformatics community. And the goal of this is to make it a lot easier for people to curate these global scale data sets or large integrated data sets, whatever your question is, as well as for me to get to myself to a curated global data set of soil whole metagenome samples. And the other things that you can imagine doing with this sort of data discovery tool is seeing where we are as a sequencing community, how many metagenomes are there, how many transcriptomes are there, where should we be focusing our sequencing priorities. The next step is that let's say I have my global data set. I then need to be able to compare these apples to apples. So I need to map all these metagenomes to some sort of gene reference so that I have a count of genes across all of my sites that I can use for then doing this functional structure discovery down the line. And in order to do that, there's no real there's no real gene reference available that's specific to soils. So I'm proposing to develop a global soil gene reference catalog, which is going to involve doing a massive co-assembly of all of these 3,000 or so metagenomes, along with the known soil genomes, of which there are 900 some. Assembling these all into contigs, doing prediction on these contigs, and filtering that into a non-redundant global soil gene reference set. Which is a pretty major undertaking. Ultimately, I'll then map my metagenomes to this to get my gene count matrix across sites. And that'll be useful for my scientific questions, but I'm calling it GISGR, the Global Soil Gene Reference. But what you can use GISGR to do, beyond my own scientific questions, is it will be a useful tool for anybody who wants to do a soil comparative metagenomic study, of course, but also you can answer some pretty fundamental questions about soil microbial diversity that we haven't really answered definitively, like how many genes are in soils total? Have we sampled all of them? And if not, how many are left to be sampled? And how does that compare across different biomes? And again, that can help us to shape our sequencing priorities. So I've got my global data set, I've mapped this to GISGR and gotten my gene count matrix. And then we then want to learn the biogeochemically relevant functional structure inherent in microbial communities. So I'm proposing to do this using a semi-supervised topic modeling approach with biogeochemically relevant labels, and that will allow me to cluster genes into biogeochemically relevant topics or functional clusters. And I can then use this learned structure to identify functional ecotypes within the global sampling sites. And also to answer questions like how functionally homogeneous are soil communities within a particular biome or globally. And then you can use this learned structure to make some useful predictions about microbial communities and how they fit into the world. So I'll then use this structure to model the relationship between microbial communities, environmental variables, and biogeochemical activities using these learned clusters. And this can answer questions like how will microbial community composition change through time? What is the sort of core metagenome that defines a functional ecotype that I've defined in step 3? And if microbial community compositions can be predicted by environmental variables, How can rates of biogeochemical processes be predicted from functional clusters? And then you can imagine really getting to a point where we could conceivably incorporate microbial information into something like an system model. So I'm going to talk about each one of these steps in a little more detail, and let's start with MetaSeq. So I imagine everyone here has some kind of scientific question, whatever your own scientific questions are, where you'd love to ask it if only you had the data set, if only you had all the subsurface transcriptomes, or all of the hydrothermal vent 16S surveys that have ever been taken, or all the soil metagenomes from prairies with an average read length between 400 and 700 base pairs, or something really specific, like you want all the bacterial genomes from alpine lakes in temperate environments where beta proteobacteria was somewhere in the keywords. And that's something on the level of the specificity that a lot of people want when they think about curating a data set, but it's really hard to actually go about doing this search. So you hit this bottleneck at the very first step, where the very first thing you need to do is curate this data set, and you don't care that much about curating the data set. You want to get to your scientific question. But you run into, you have to know all the APIs for all the different repositories, you have to be able to search and filter on all these things. They don't make it very easy to do that, so it requires a bunch of skills that either you may not have, or you may not want to learn, or you may not have the patience of doing, even if you know how to do it. And this is because sequencing data is spread across a lot of different repositories. There are metadata standards to describe sequencing data that most of these repositories use, but the way that repositories expose it to the user is really variable. And it often makes it not searchable, even if they do have this metadata, they often don't make it very searchable for anything beyond the basic, like, oh, I'm looking for metagenomes. That's not really very helpful. So if you wanted to do like something like this last query, you couldn't really do that with existing resources. So MetaSeq is designed to take the work out of the data discovery process, bringing together metadata from all the major sequencing repositories, cleaning up that metadata into the MetaSeq server, and then presenting that to the user in an interactive, really lightweight web app, so that a user can really easily explore, search and filter on data sets, and get to that curated data set a lot faster and more easily. It also allows you to save a curated data set, share it between different MetaSeq users. And the last thing that I think is really important is it encourages improvement of the metadata pool. So the metadata pool right now, the metadata sort of describes all the sequencing data. Even though there are metadata standards, they're really not well controlled, and they're just riddled with errors, they're riddled with missing fields. And I could get on my soapbox and talk all day about how we should be doing more in metadata ingestion, but this is a really rich data set that has a lot of information in it, and I think there's a lot more we could be doing to correct errors, improve the metadata pool so that our existing data is more useful. So one of the, I think, hardest steps of MetaSeq is the metadata cleaning step. It's the very first thing. What I'm essentially doing is it goes to all of the different sequencing data repositories, and it uses their particular APIs to get the metadata from every sequencing sample that they have in there. And it tries to boil down that metadata to adhere to the MixS standards. The MixS is the name of the metadata standards that the Genomic Consortium uses, and that most of these repositories use. And it tries to sort of harmonize that to the MixS standards, because often you'll find that even though they're supposed to be complying with this, they don't really. You'll have, for example, column names that are supposed to be environmental package, but users have been able to sort of enter whatever it is, specific thing they want. So it tries to boil all these down into the same column name, so they're all unified. And it also tries to harmonize across repositories. So for example, NCBI uses nbiome, whereas MGRAS uses biome, and you want to make sure that these are all going to end up at the same place once they get into MetaSeq. It also does some controlled vocabulary parsing. So for a lot of these metadata fields, like investigation type, for example, you're supposed to enter 1 of 8 fields and only 1 of 8 fields. But because it's free text and you can just kind of enter whatever you want, people enter all sorts of nonsense. So you need to find a way to try and parse all of this into the actual control vocabulary. So when you're down the line trying to search on bacteria or archaea, you don't miss some because they've been mislabeled. So all that metadata cleaning step happens, and it ends up getting into the MetaSeq database, where you then serve up that data if somebody's added some search or filter parameters, serves it up to the user so that they can visualize this data in a really lightweight way. So one of the major challenges of this is if you're looking at 2 million samples, you don't want to just send all the data for those 2 million samples to the browser because it'll crash immediately. So you have to be a little bit clever about how you send data. You send a summary of the data, like the heights of histogram bars, and not the actual data itself, stuff like that. And then this stuff is visualized right now in a React web app that allows you to really rapidly add filter parameters and things so that you can visualize data in a really lightweight way, explore it really easily, and get to your curated data set faster. So it is in existence, although you can't get there unless you're an admin right now. But you can sign up to be a beta tester on metaseq.cloud, so please do if you're interested. And you end up going to the main Explore page, which lets you search and filter. Here I am logging in with my Gmail address that I can save a discovery. And then there's some search and filter parameters here. And when you select one, everything sort of automatically propagates, and you get this sort of interactive visualization of your filter parameters. And one of the things I'd really like to do is to make your filter parameters responsive to the kind of data that you're looking for. So if I put up in a different filter parameter that I'm looking for a genome, you might care about filtering on different things for a genome than you would for a MIMARC survey. So you be a little bit responsive to previous inputs to make that a little less overwhelming. And then you can see a lot of interactive visualizations that you can do to sort of explore the curated data set that you have so far, see if there's any artifacts in it, or get ideas of like, oh, well, maybe I should filter out any average read lengths below 100 or whatever it is for you. And then at the end, you can save your discovery to your profile, and you get like dummy code for data download. So if you're looking at 3,000 samples, you'd probably not want to just run this code, but you could, if you were just downloading 10 samples that you've curated, you could just run this code and it would download it, download the original sequencing data for you so that you could go forward with your actual project. You also can browse other users' discoveries, save ones you like, or use one as a launching off point to add some more filters to it. And the last thing is that you can improve on the metadata pool. So either users or MetaSeq itself can flag errors in metadata. You can imagine doing this automatically, like during latitude and longitude parsing. For example, MetaSeq will try and parse the user inputted latitude and longitude, which is a text field, into a number. But you can imagine that there's lots of problems with doing that. And if it can't do that, it'll be like, well, I know something's here, but there's an error. So you can do some automatic error flagging. You can also have a user, when you're looking at metadata, sometimes it's really obvious that there's an error. So you could flag it. Sometimes it's really obvious what the correction is. So you can submit a correction if you can tell from the context what it should be, or maybe you're an author, or maybe you know what the paper is and you've read it. So you can have this sort of user annotation of data sets or discoveries, and user correction of metadata errors, so that you can imagine that over time our metadata pool could get better, which would mean that searching and filtering on the metadata would also get better. So MetaSeq, it's super lightweight because it's built completely on the metadata and not the sequencing data itself. So you can have 2 million data sets represented by MetaSeq for a few gigabytes on the database, which also means it's pretty cheap to maintain, which is great. So depending on web traffic, I think that it'll be something between 30, 150 a month, which really isn't too bad. So I anticipate that the beta version will be released in a couple months or so. So if you are interested in this, please do go to metaseq.cloud and sign up, and I'll email you when I'm ready for beta testers, because I'd really love to get everybody's input. So that's MetaSeq. Using MetaSeq, I'll get to my global curated data set of soil metagenomes. And using this, I want to create my global soil gene reference. So essentially what I need to do with this is take my 3,000 metagenomes and all of the genomes from soil communities that we have, which are conveniently packaged in a thing called RefSoil for me, of which there are maybe 900 or so, and then massively co-assemble all of these so that I can compile all these gene fragments into longer contigs. On the contigs, predict genes, and then take the predicted genes and cluster them into non-redundant sets, so that you ultimately get to non-redundant gene reference of soil genes. But as I mentioned earlier, there's a lot of computational barriers to doing this. So with a smaller sample where assembling your metagenome and predicting genes on it is really pretty reasonable, once you get to 3,000 samples, that gets really overwhelming really fast. So MegaHit, which is one of it's the fastest assembler that I know of, can assemble, for example, a 250 gigabase pair data set in 43 hours, a 484 gigabase pair data set in a little over a week in reasonable memory requirements, under 500 gigabytes memory. But I'm really talking about assembling like terabase pairs or petabase pairs. And these don't scale linearly, as you notice. So that means that doing the co-assembly, you might have to do something a little clever to make your assembly more reasonable. Like breaking up your assembly, maybe you have some metagenomes that have no overlap. So you could do, instead of doing one gigantic assembly, you can do 10 or 20 assemblies separately and then sort of compile these at the end. But I would argue that it's really worthwhile doing this because if you map directly to KEGG, for example, or one of the other sort of curated database or annotation databases, with soil metagenomes, you typically are only going to be able to annotate something between 10 and 30% of your genes. So you're going to be throwing away a ton of information, and inherent in that is a lot of that functional structure that we want to pick up on. So it's really worthwhile to have a common gene reference resource for soils, both for myself and I would argue for anybody doing soil comparative metagenomics. So one way that we might be able to do this massive co-assembly is reduce our memory requirements by partitioning our data sets. So this is an example from a tool that Tez Brown has called Sour Mash that allows you to sort of partition your data sets into different hash groups that and compute the similarity between your data sets really quickly so that you can identify maybe groups of data sets that have essentially no overlap with anything else. So you can partition your all these data sets into a group of what looks like, I don't know, maybe 10 subgroups and do your co-assembly separately on those groups. So you could really decrease your memory requirements and the amount of time that you have to run it by doing that. You can also imagine maybe reducing your data size. So probably there's plenty of reads where in the metagenomes where we have them in the RefSoil genomes. So why would we increase our data size by trying to assemble all of those? Maybe we could just throw them out. So you can imagine maybe mapping your metagenomes to RefSoil as a sort of pre-assembly process, and anything that maps with really, really high similarity, just toss it out and do your assembly with with the remaining sequences. So then once you have your contigs, you need to do de novo gene prediction. And a paper that I thought was pretty convincing argued that you want to do that you should take the consensus of multiple de novo gene predictors for short reads, for short read sequences. And then you want to cluster these genes into a non-redundant data set to get your final what did I call it? Global soil gene reference. And then you can annotate GISGR to KEGG so that you do have KEGG annotations for any of those genes that are of known function. So then once you have GISGR, you can map all your metagenomes back to this gene reference, and you ultimately will get what we want for actual topic modeling input, which is our gene count matrix. So for every metagenome site, we have a count of all the genes in GISGR at that site. So I would also argue that GISGR is useful for scientific reasons, in addition to computational ones. Things like you can imagine making a rarefaction curve out of your gene set and trying to predict how many genes there are in soils. Have we seen the amount of genes level out here so that we think that we've actually captured the full diversity of soil genes, which I sure hope that you would with 3,000 samples, but it's not impossible that you wouldn't if you have bad assemblies. Or are we seeing things not converge? And in that case, you can then project this curve out and predict, well, how many genes are there left to be discovered? And you can imagine breaking this down by biome. Maybe some biomes are more well sampled than others. And maybe we should focus our sequencing priorities on those that are less well sampled, for example. Okay, so we have our gene count matrix. And now we can use this to discover our biogeochemically relevant functional structure in microbial communities. So the way that I'm proposing to do this is with semi-supervised topic models. What topic models do, they originally developed in the text mining community, but they essentially they presume that every metagenome in your site is represented by a mixture of latent functional topics, and that every topic is represented by the relative likelihood that it's going to generate that gene. So if you have one topic, it might be really likely that you draw one gene out of the hat, and another topic, it may be more likely that you draw a whole different kind of gene out of the hat, right? And every gene in the metagenome is there because it was generated by one of these topics that are represented that are more or less likely to be found at that site. Yeah, let's leave it there. So the way that it does this is it essentially iteratively goes through every gene and every site, and it assigns a topic assignment for that gene at that particular site. So it takes into account both the sort of local frequency of the gene and the global frequency of the gene. So it goes through gene 1 at site 1, gene 2 at site 1, all the way down through site 1, then it goes to gene 1 at site 2, gene 2 at site 2. And it assigns a topic until you reach convergence on your site-specific gene topic assignments. And this produces ultimately 2 really useful outputs. So from that you can derive your set of topics. So in this example, I'm only looking at 2 topics, and the likelihood that if I had a bag of my Gisger genes that I'm going to draw gene 1 or gene 2 out of the hat in topic 1 is going to be different than in topic 2. And then there's also the site topic distributions. And that's really useful because a particular site is going to be made up of different proportions of the different topics. So site 1 here is more heavily related to topic 1, site 2 to topic 2, and site 3 is kind of a mixture of both. So what I've really described is an unsupervised learning structure where you're taking functional data and you're sorting it into different structures, but you're not using anything that you know about the genes to do that clustering. So this is similar to k-means or something if you've done that. But what I'm really interested in doing is discovering biogeochemically relevant functional clusters. So in order to do that, there's a semi-supervised extension of topic modeling, which essentially means that I'm going to take all of the unlabeled genes that I have, and I'm going to still use that to try and discover the structure in my data. But I'm going to use labels where I have them, annotations where I have them, to try and guide the algorithm towards clusters that are more useful to my goals, in this case, biogeochemically relevant clusters. So what I can do is I can take in Gisger for all of the KEGG annotations that correspond to different components of the nitrogen cycle, I can label those genes as like, well, let's allocate 16 topics to the nitrogen cycle, and I'll let the algorithm sort of figure out how to organize all of those within that group, but I'll sort of restrict it to this set of clusters so that I can hopefully sort of partition genes into biogeochemically relevant. So when you're going through this process where you're making topic assignments, in this case, if you have an unlabeled gene, it's going to do what it normally does. It's going to assign it to any one of the topics. If you have, say, a nitrogen cycling gene, or I've allocated a different set of topics to carbon cycling or whatever it is, I'm going to say, well, you can only really assign it, for the most part, to topics 1 through 16. So it's going to make topic assignment within those restrictions rather than through the whole level of topics for only those genes that I have labels. So the way that I'm thinking about doing this is using all known genes involved in biogeochemical pathways. So in this example, I'm looking at the nitrogen pathway or the nitrogen cycle and giving it a range of topic possibilities. In this case, if there are 8 main sort of processes that you're looking at, say, let's double that, let's give it 16 topics it can choose from and let it sort of learn the structure within that. So that they're a little bit flexible about, because some of our things like the carbon cycle where we have really bad annotation and like all the processes, there are so many processes, I'm not going to break them down super well. I can give it a little bit of flexibility to like discover the structure within this general guideline. And the other thing I can do is make soft assignments. So instead of saying you can only be in these 16, I can say like, you can pretty much only be in 16, but like if you really, really want to, you can be outside it. And do this for all of the biogeochemical cycles that I'm interested in. So carbon, sulfur, phosphate, micronutrients, all of the biologically active elements. So we go about learning this functional structure, and we end up with some kind of set of learned topics, and then we can look at these topics to try and derive, try and interpret the functional structure that it's learned. So one thing I'd like to do is sort of break down the sites here. So here I'm just sort of visualizing all the sites and their relation to all the different topics, and the thickness of the line has something to do with, is proportional to its topic weight. This is made up data. But, and you can imagine trying to define within this structure a set of functional niches or ecotypes among sites. So as an example here, like here at site 3 and 6, like they have a lot to do with topic 5. They're kind of here away from all the other sites. They don't have a lot. They have a little bit of topic 3 and pretty much none of the other ones. And that's really distinct from other ones. So I might decide that that's a functional ecotype. And you can use network analysis approaches to try and quantify this. Or you can also imagine looking at the relationship of topics to genes, where some genes are really specific to topics. They have a really high weight in one topic, but not really present in other ones. Or there's other genes that are really cosmopolitan. So you can imagine using that to try and make hypotheses about gene interactions. So maybe you have a topic that's like all carbon cycling genes and then a couple of nitrogen cycling genes. And that maybe tells you something about like, well, maybe there's an interaction between those. Or also to make hypotheses about function prediction, for example. So like if you have a lot of carbon cycling annotations and then a bunch of genes that aren't carbon cycling annotations, but they have really similar topic distributions, that might be an indication that they have similar function or that they interact in some way. And then you can also look at things like the connectivity in this network, so how related every site is to every other site, to think about how functionally heterogeneous are soils. You can look globally, or you can look, within particular ecotypes. Some ecotypes may be really homogeneous, some less so. So then we can use this learned functional structure to make predictions about microbial communities. So what's really cool about these topic models is that they're generative. And what that means is that all of our sites are now represented by a probability distribution of all these topics, and each topic in turn is represented by the probability that you draw a gene out of the hat. So you can then, given this probability distribution, simulate a metagenome for a site with a particular topic distribution. So you can predict microbial community composition given a set of topic distributions. So if you could predict topic distributions really well, you could predict microbial community composition conceivably, or you could also use this like as a validation step. So you can simulate a metagenome where we know it has a predicted or a derived topic distribution and see how similar it is to the real metagenome. And if it's really similar, that's an indication that you have actually captured the structure of that, of your microbial data sets, right? But one thing that I think this can be really useful for is in your identified functional ecotypes that we identified when we were doing the analysis of the functional structure, you can then use that to predict like the average ecotype metagenome. So you take all the site distributions of those sites that make up an ecotype and you generate a bunch of metagenomes, given those site distributions, and it takes with the average consensus metagenome. And now you've made a prediction about what makes up this ecotype at a genetic level. And then we're really interested in how can we predict these topic distributions and predict microbial communities from environmental data so that you can make global projections of either topic distributions or of community composition itself at a global scale. So my plan is to do some multivariate regression models using environmental data that's available via satellite so that you can model topic distributions. And if you can get pretty good accuracy of modeling the topic distributions, you can then use the predicted topic distribution to also predict the community composition at a global scale, which is really cool, I think. So that's the general plan. So just to summarize a bit, the real goal here is to uncover the underlying functional structure of soil microbial communities with a novel semi-supervised topic modeling approach. I don't think topic models have ever been applied in environmental metagenomics, so I'm really excited about seeing if it's a promising way to go about it. And in addition to soil metagenomes, I'm planning to do a proof of concept with marine systems as well. So I have to do, I have to finish up MetaSeq, I have to do GISPR. So in the meantime, I can be working with the TAR dataset, for example, like a small, pretty like well-packaged marine dataset, map that to the OMRGC, which is the marine gene reference set that the TAR people came up with, and use that as a proof of concept to sort of go forward with the topic modeling in the meantime while I'm doing all this GISPR and MetaSeq development. So if you're interested in the marine stuff, you'll see that as well. And then I can also use this to identify functional ecotypes and hypothesize about important genes or interactions between genes, analyzing the learned functional structure that we've identified. And then we can also use this to model and predict global patterns in microbial functional biogeography. So if we can model the relationship between environmental variables and functional composition, we can get a lot closer to integrating microbial information into Earth system models. Also predicting like core metagenomic makeups of functional ecotypes and predicting microbial community composition or topic probabilities at a global scale. It also produced a global soil gene reference catalog from a massive co-assembly of global soil metagenomes, which is having a gene reference like this is really a prerequisite for comparative metagenomics, and I think there's a growing interest in doing this kind of thing, so it'll be a really useful tool for myself and for the scientific community. But it also allows us to address some fundamental questions of soil microbial diversity. And finally, to develop a data discovery tool for future integrative studies, and for my own purposes, to curate a global scale soil metagenomic data set, facilitating data discovery and integration for comparative bioinformatics in general, and for comparative metagenomics specifically from my perspective. So with that, I'd like to thank my advisor and my committee members who have given me a lot of really interesting, valuable input, and my co-authors, Nick Brown, who has helped a lot with the MetaSeq development as well. With that, I can take any questions.
So, questions for you?
Do you have a plan to deal with data quality, like depth of sampling and sample handling and sequencing method, all those kind of questions?
Yeah, and it's really difficult is the short answer. And there's not, what makes it especially difficult is that there's not a lot of like community consensus on what exactly you should do right now, I would say. So there's one paper in particular that I found really useful that Katie Pollard and her graduate student Stephen Defetch published last year that it has comparative metagenomics in the name, and it's about being able to integrate these data sets. And they talk about all the different challenges that you need to look at. Like one of the things that I thought was really interesting is that they did some simulated experiments and decided that things like rarefaction aren't that important, which is really potentially useful when you're doing comparative study where you have a large variation in your sequencing depth. So maybe you don't have to worry about that so much, and you can just use that. Of course, you'll need to normalize your gene counts and everything. So yeah, I mean, it's a very difficult question, and one that I will have to deal with.
I mean, I can imagine some of the relevant stuff being in metadata, but I don't know how you deal with it after that.
Yeah. So you can potentially like filter out a lot of your stuff at the metadata level, assuming your metadata is correct. Yeah, like you could filter on your average base pair read, for example. One of the things I'm really worried about actually is that because there's a lot of metadata errors, you filter on, you say like, I want a metagenome, but drives me crazy. 16S surveys are not metagenomes. So people will mischaracterize 16S surveys really, really frequently as metagenomes. So you do get to sort of this point where you do kind of have to do a manual curation, like even with MetaSeq. So that's something I'd like to see you have to do less of in the future, but you do have to like really watch out for that.
So why the need for like the whole scale assembly? Like why do you think that would actually improve assembly versus having sort of just the smaller assemblies? I mean, that's a lot of computational power to be able to do such a broad assembly. But with, there's also pitfalls in that, in creating a lot of chimeric genes.
Right.
So I'm just confused about why.
Yeah, I think one of the things is that soil metagenomes don't always assemble super well, and if you do a co-assembly, you might get better assemblies. Because you might get overlap in two metagenomes where you wouldn't have gotten it in a single metagenome because they're rare genes or whatever. So that's one answer. I also think you really need that gene reference. I don't know if that's exactly what you're asking, but
yeah, it seems like for the maintenance. So it's every time libraries are added, does the assembly have to happen again?
No, I don't think so. So conceivably, if you sampled enough samples that you sampled the full diversity of genes in soils, you shouldn't have to do any more sampling, right? Like if you've gotten the full diversity of samples, you should have a gene reference set that's representative of the entire globe of soils. So if you went out and took a sample and did an assembly and predicted genes on it, they'd be genes you already had. They're not adding any more information. So that's kind of what the rarefaction curve that I was showing is. You hope that you get to the level out one and not the not level out one. And once you've gotten to that,
in soils, is that the case? Because it's definitely not the case in the marine environment, right? I mean, we're still on
RGC, it was pretty well sampled.
Mm-hmm.
And that was with a relatively small number of samples. It was 200 or so. And soils, you would imagine, are more diverse. So you probably need more samples. How many samples is, I don't really know.
And so you mentioned about also the maintenance. That's a huge undertaking. Is that something where as soon as you get this up and going, hopefully, how do you maintain it with all for MetaSeq? Yeah.
So from a financial perspective, you could imagine carrying it pretty easily because it's a couple thousand. What's harder is when there's a bug or you need to address an issue or the server's down or whatever. You have a problem there. So one thing is that the scrapers, all the metadata scrapers and everything, are fully automated, so you can have the updates to the MetaSeq database happen on a weekly basis automatically. So the INSDC updates every day, and you can have MetaSeq just okay, give me all the new ones, scrape the metadata, stick it into MetaSeq. So the first thing is just automate as much as you can, and then it'll be an open source project. So the hope is that I'll maintain it, but also if you want to, there'll be an API that you can use to query MetaSeq, or also to build on features if you want to plug MetaSeq into some other tool you're building, or potentially down the road integrate it with some other tool that has more infrastructure resources.
Okay, so from a very basic understanding of what you're doing, Adrienne, I think you're going from metagenomes to predicted biogeochemically relevant functional clusters, then maybe predicting out of that the community structure, but then going in the other direction, kind of taking it downstream. Could you maybe comment on ground truthing some of those functional clusters that you've identified? Yeah.
Yeah, so model validation is super important. So there's a bunch of stuff that I think you could do. One of the things I mentioned was you have your predicted topic distributions and you generate a metagenome out of that. But you also have the original sample, the original site, you know what the true metagenome is. So you can do that for all of your metagenomes and compare it, like how similar are they? If they're really similar, that's an indication that you've actually sort of captured the functional structure of your real community in your topics. If they're not so similar, then you have to ask why. You can also think about like trying to cross-validate a little bit. So you have, when you go into your, when you have your biogeochemical labels, you have for all the genes that are annotated, that biogeochemical label that you've given it. But then you can hold out like say 10% of those genes where you would have labels. You can just like hide the label and treat them as unlabeled genes and do your topic learning. And at the end of it, you should, if you're actually sort of picking up on the biogeochemical labels that you want to be finding, you can then uncover the label that you actually know like what you want your label to be and see, was this gene ultimately given the topic weights that I expected? So that's an indication that like, okay, these are actually going towards the biogeochemical direction that I want. You can also like sort of look at a very broad level, like do you see in a topic where you have a lot of nitrogen fixation genes? Do you see that topic being at a higher weight in like a plant-associated community as opposed to like a really nitrogen-rich like forests or like jungle community? You'd imagine to see like those sort of expected outcomes where you'd expect your topics. If you look at your genes that are in your topics and they sort of seem to be going in a certain direction, like, oh, well, I'd expect this topic to be really like heavily weighted in a site where that's relevant and less so in a site where that's not relevant. So you can sort of at a general level do that. And then we talked about committee meeting about doing some like clever stuff where if you where you could potentially test computationally, like, do these genes co-locate on genomes? If they're associated with topics together, do they also like co-locate on genomes from Resoil or whatever it is, or from your contigs? And then I would argue that if you get better predictions out of your topics than you get with like genes or species like previous models have done, that's also an indication that you're onto something. So yeah, I think the key is to just come at it in a lot of directions. And if all of those point in one direction, then that's a good sign.
Actually, I have a question. Why do you want to start with soils? Because they are, so is my worry, biogeochemically particularly complicated. Soil particles, if they are not completely waterlogged, harbor an immensity of small-scale chemical gradients really on a microscopic scale. Within a tiny particle, you can go from aerobic to anaerobic and back. Whereas when you are in a, let's call it aquatic sediment, you have often more homogeneously structured sediment, where the fact that everything is embedded in water imposes a greater predictability of, for example, redox change. And that, of course, should have implications for generating microbial predictions about gene content, predominant pathways, etc. Seems to me that once you have a system completely set up and going, the very last environmental habitat where you want to unleash it, when you are completely sure that everything is working, the hardest nut, so to speak, that has to be cracked would be soil. But in the construction phase, when you are dealing with all kinds of problems of implementing this, ground truthing it, etc., aquatic sediments seem to be a little less complicated and might give a more manageable test environment in order to get the whole thing going.
Yeah, that is something that I have questioned as well. My main reasoning for it was that there are just so many more soil data sets. So then it becomes a question of like, well, how much more complicated are soils than marine systems or sediments? Are they, more than 10 times, it's something like 10 times more soil samples than marine samples, for example. Are they more than 10 times as complicated, or are they less than that? So I think that's hard to answer. So my main reasoning is that there's more, I don't know where that is anymore, but it doesn't matter, that there's more soil samples than any other kind of sample. And I also think that it's a really, it's a area of interest for a lot of people who are interested in things like environmental predictions and things as well. Especially when it comes to sediments, there are even fewer samples. Is this it? Yeah. There are even fewer samples than marine samples. They're like, I think like 100 or something like that. Not a lot of like waterlogged sediments. So if I were to not do soils, I think I would lean towards doing marine water column. And I think that you could argue that I should. I think that particularly as I do the like Cara data subset of stuff, that I'll sort of be able to tell if that's like a really promising direction to go in. And I would argue that the history stuff is useful regardless of whether or not the topic models worked out. So I could then, see my initial topic model results from soils and use that as feedback compared to like my marine Cara thing and decide from there if there's like, I really should abandon this soil idea because it's not.
No, when you have marine environments, you could regard the water column simply as a particularly extended version of the benthic boundary layer. It has oxygen. So you divide your marine sample sets into the oxic water column and the anoxic deeper layers. They may be sediment, they may be stratified basins. And you have some large-scale geochemical gradients and controls, which are fairly easy to control.
Yeah, I'd want to, if I did that, you'd need to incorporate, I think, sediment genes into your gene reference. Because even though you, from a geochemical perspective, that you could maybe argue that the like community compositions are really different. So I would imagine you couldn't map to the OMRGC without accounting for that somehow. But you could maybe account for that somehow.
Okay. Yeah, no, I would still be attracted to the marine metagenomes, simply because I think they are, on the whole, they give you more bang for the buck with respect to geochemistry. In soil science, I think many soil parameters there are historical parameters, color, crumbliness, etc., etc. How do you want to connect that to microbial community structure?
Yeah, well, the environmental stuff, I'm planning on using all satellite data anyway, because what I really want to be able to do is project it with water spatial skills. So that, you have pretty basic stuff in both marine and soil.
I mean, when you think about all these things considered, I mean, time is working for you because more and more marine metagenomes are popping up sooner or later. So the difference in numbers will become less relevant in time. So maybe you should think about it again. I don't want to impose anything, but I think the marine metagenomes give you a greater level of more handles really to get at substantial biogeochemical information, which is quantifiable in a chemical sense.
That is a valid criticism.
I was going to say, why don't we have more questions from non-committee members?
I was just going to concur with Andreas, and I actually think it could be an advantage that you have a much smaller database to work with. Because it seems to me a lot of the challenge here is how to scale up and quality control and integrate such enormous databases. So if you start with something smaller, you learn a lot of things about it that then you could use those lessons later on when you're scaling up something bigger. But the other advantage to the marine environments are there are a number of really well-studied environments where the biogeochemical structure is really well known. I think like, well, Cape Lookout Light's a good example. And there's Long Island Sound, there's Aarhus Bay, just where, if you look at centers of marine biogeochemical research and you look out their back doors, you will find spots where rates, the structure. And so it's a great place to do some proof of concept, which is sort of my other question or comment is, even if you have perfect information about all the genes, there's still a challenge to put that together into a structure and into a rate. In terms of the structure, there are facultative organisms, so I imagine there's some ambiguity from their genes as to what they're doing in a given spot. And then in terms of, like, you think of the rates of, say, even photosynthesis, it's not just the genes, but it's the nutrient concentrations, it's the light availability, it's the stratification. For sulfate reduction, it's the rate that the substrates are being provided. And so it'd be great to do some proof of concept before getting into this really giant data organizational problem to show that how you connect the genetic information up through the environment. So you pick these, like even for the soil environments, these LTER sites, there must be some great data there where you just focus on one site and just show that it all works. And then you say, now it's worth going through and putting everything that's ever been done everywhere together in this thing that we can ultimately put into a global model. It'd be nice to have it ground truth at one or two sites, say, to show that it works. And then you put all that, sounds to me like months, if not years of effort to dealing with all of that. And the quality control that Barbara was talking about, et cetera.
Yeah, and one thing I talked about in my written proposal, but I don't think I mentioned, is I would really love to take a stab at trying to predict rates, which we're really bad at doing from genes. But if you were to take all the environmental data that you have and all of the topic learned stuff that you're using and bring that together and try to predict actual rates of like respiration or whatever it is. And yeah, I think you could do that in marine and soils as well. The LTR is a good suggestion. My plan was to just like try and get it from a bunch of different environments, but it might be a good idea to just take one subset.
Yeah. And I do think the rates are no better in the marine environment for the same, just soils are so hard. Like you look at the Duke Forest, it's like every little spot is completely, they have little flags and they're completely different because something's growing there, something else is growing there. Marine, it's I don't want to say it's completely homogeneous, but compared to the soil environment and the fact that it's always wet is really nice.
Yeah.
It's so stable too. I mean, you don't get the same swings of environmental parameters that you do in the soil. So, it just seems like it's 1,000 times harder to make this work for soils. Right? So why not start on the easier problem?
Yeah.
But you like tough problems.
An easier question. I like soil, but anyway, I think soil is cool. But one is, do you really think it's necessary to make a separate set of reference genes for each environment, or rather, I think there's a lot of overlap among them? And then the second one is, how do you deal with updates to annotation as more genes become discovered and described and so on and so on?
So you mean overlap between gene references like the OMRGC and like GISCR? Between like marine and soils? Is that what you mean? Or like?
Yeah, for your annotation step, you're going to create a set of reference genes for this environment, that environment. So I would think there's a whole bunch of overlap, like all the DNA genes, ribosomal genes, blah, blah, blah. So I don't know if it's really necessary, or do you think it's necessary? I guess.
I think you can often, because you do, but the way you map it, like you have a higher danger as you expand out of getting mapping to things that are not really right, where there might be something that's better over here, but you weren't looking over here, you're looking over there. So that's one issue. I think that you probably could. One of the things that I would do after producing GISCR is sort of compare my gene set to like the OMRGC, or the Tara folks, when they made the OMRGC, compared it to like human gut metagenomes, I think, and sort of analyze the overlap. How different are they? And then you could conceivably, I think there is some overlap with like ribosomal genes and things. But there's a lot of differences too. So I think it might be worth having different sets. But I could imagine down the line wanting, for whatever reason, to have a unified gene set because you are now making like truly global models of microbial communities where you're incorporating
Working at an
terrestrial and marine
interface,
systems.
Internetary, you're going to have maybe
Yeah,
some
or in the
overlap.
Yeah.
Anyway, and then the other question was, how do you deal with, as annotation hopefully proceeds, it gets better.
The annotation part isn't the computationally hard part. So I think you could just re-annotate it. Just recompare you could subset out all the ones that already have annotations to like have to not, well, actually, maybe you wouldn't want to do that, but you could just re-annotate it to KEGG or to COG or whatever it is that you want to annotate it to over time. Does that answer your question?
No, I think that has to be a quality control step at some point, but that's for the future.
You mean quality control for
annotation, just well, I guess by waiting for it to get into KEGG is one way to do it, but a lot of things take a long time to get there.
Yeah. Yeah. And I think, so for Gizker, I mean, you can annotate to all of the references that you want. And then a user can decide what level of, like, do I want to look at KEGG annotated stuff or do I want to look at stuff that's maybe a little bit more flexible? And then, you could let the user sort of do that downstream, whereas Gizker could be like a more general tool.
Any questions from IMS? Okay.
One of you. Thanks for coming.
All right. Any other questions from the audience? The committee will stay here for further questions. If not, thank you.